Welcome to This Explainer. Today, we're diving into something that sounds straight out of a sci-fi novel, but is actually happening right now. We're looking at a massive, groundbreaking milestone in reproductive science, and, oh boy, the profound ethical minefield it immediately creates. It's called Revolutionizing Reproduction, the Science and Ethics of IVG.
So imagine this for a second. What if a routine blood draw was literally all it took to create a human egg? Sounds totally impossible, right? Well, a Berkeley company named Conception recently announced a huge leap forward in making exactly this a reality. It's a breakthrough that's fundamentally challenging everything we thought we knew about the absolute limits of human reproduction.
Let's get right into it. Section 1. Blood to human eggs, the breakthrough.
Get this, researchers have actually successfully grown the earliest human eggs ever made entirely from stem cells. It's a world first. The technique making this happen is called in vitro gametogenesis, or IVG for short. Basically, IVG is the generation of mature sperm and eggs from ordinary adult cells, like your skin or blood cells, entirely within a laboratory setting. It's literally opening up pathways to procreation that we used to think were biologically impossible.
The biological process here is just an absolute marvel of cellular engineering. It starts off so simply, just a routine blood draw. Then, those adult blood cells are reprogrammed. They're essentially put into a cellular time machine, rewinding their biological clock all the way back to an embryonic state. From there, scientists carefully coax them into becoming primordial germ cells. And finally, by combining these with helper cells, they form lab-grown mini-ovaries. They are literally building the exact biological structures needed to mature an egg, all sitting right there in a petri dish.
Which brings us to Section 2, the 10-year gap between mice and humans.
Believe it or not, the mouse version of IVG actually arrived a full decade ago. Yeah, 10 whole years. Scientists successfully used mouse skin cells to create eggs that produce perfectly healthy pups. So, you might be wondering, if the proof of concept existed a decade ago, why on earth did it take 10 years of agonizingly intense lab work just to reach this early milestone in humans?
Well, the answer really comes down to timing and extreme delicacy. See, a mouse only needs a few weeks to develop germ cells, but humans, we require months. The human ovarian environment is this incredibly slow, finely tuned, delicate ecosystem. In trying to rebuild that precise, long-term nurturing environment in a sterile laboratory dish, it turned out to be exponentially harder than anyone ever anticipated.
Moving on to Section 3, the promise. Expanding reproductive possibilities.
If researchers can perfect this, IVG offers just staggering new possibilities for folks who might otherwise be completely unable to have biological children. It is a potential game changer. We're talking about helping patients with complete infertility or those without ovaries. It completely opens the door for same-sex couples to have children genetically related to both partners. Like, imagine creating eggs from a male's cells. Plus, it could preserve fertility for prepubescent cancer survivors, completely bypass the natural decline of aging, and even allow for large-scale screening to prevent severe heritable genetic diseases. It's huge.
But we do have to talk about Section 4, biological and safety hurdles.
Because long before this becomes an actual clinical reality, the American Society for Reproductive Medicine, the ASRM, is warning us about some massive biological barriers. Right now, to be totally frank, IVG is highly inefficient and carries some severe risks. In mouse models, the live birth rate is a remarkably low 1 to 3%. That's it. And worse, the very act of reprogramming these cells carries huge risks of epigenetic errors. That means a high chance of unexpected, dangerous developmental abnormalities. And because the whole process relies on stem cells, there's an inherent risk of tumorigenicity, basically uncontrolled cell growth that can lead to tumors.
Because of these profound unknowns, the ASRM is stressing that without highly reliable reproducible safety data from non-human primate models, the biological risks to any future offspring are just way too great to even justify clinical use right now. Assessing fertilization, embryo development, and long-term health across generations in primates is an absolute non-negotiable prerequisite before human trials could ever be considered ethically viable.
And that perfectly segues into Section 5, the ethical minefield.
This brings us to some truly profound societal questions recently explored by Oxford University Press and the ASRM Ethics Committee. There's this incredibly stark contrast between the amazing promise of IVG and its ethical perils. Think about it. The ability to select the healthiest embryos could inadvertently lead to the commodification of human life, dramatically widening social divides. Then there's the wild possibility of solo reproduction, creating both sperm and egg from just one person, but that carries a massive 25% risk of genetic disorders. And while the idea of eradicating genetic disease sounds universally positive on paper, ethicists warn it could actually lead to decreased medical research and increased discrimination against folks currently living with disabilities.
IVG also throws some totally unprecedented dilemmas our way. For instance, there's multiplex parenting. That's the potential to create embryos using more than 2 genetic parents. There's also a real risk of society devaluing non-genetic family ties. And perhaps the most unsettling of all is unauthorized DNA use. Seriously, imagine the terrifying possibility of someone swiping your discarded coffee cup or just a stray hair follicle and using it to create a genetically related child without your consent. They could even do it posthumously, after you've passed away. It's wild to even think about.
And this brings up a crucial societal question, perfectly captured in a quote from the 2026 ASRM Ethics Committee. They state that IVG risks reinforcing genetic exceptionalism, the idea that genetic ties are essential to forming a real family. If society starts hyper-prioritizing these lab-made genetic ties, are we going to marginalize families formed through adoption or traditional gamete donation? The committee explicitly notes that this hyper-focus could disproportionately challenge our broader understanding of what it means to be a family, which could be especially tough on LGBTQ+ families.
Which leads us to our final topic, Section 6, regulating the future.
With stakes this astronomically high, proactive engagement from policymakers is absolutely 100% urgent. The ASRM insists that IVG has to be heavily regulated. Initially, any research must be limited to strict institutional review board oversight and fully comply with the International Society for Stem Cell Research standards. And paramount to all of this, we absolutely must protect this technology from premature, exploitative commercialization before it is definitively proven safe.
We are standing at a very critical crossroads right now. As the ASRM puts it, public debate and education are crucial prerequisites that should occur before society considers developing and offering IVG for human reproduction. Basically, it is imperative that society, meaning you, me, all of us, gets to decide the rules in an open, educated manner, long before the sheer momentum of this technology simply makes these profound decisions for us.
So I am going to leave you with this final thought to ponder today. As lab-grown eggs inevitably transition from theoretical science fiction into a tangible clinical reality, who exactly gets to decide the future of human reproduction? As science gives us the immense power to turn the impossible into reality, how should our society govern the very building blocks of human life? Thank you so much for joining me for this explainer. Keep learning, stay curious, and keep asking those big questions.